All right, guys, so here's another awesome lecture on this time on restriction digests. So this is another application of using rDNA. So essentially we're taking restriction enzymes and we're cutting pieces of DNA, and then we're going to use jelly electrophoresis to separate them. So we can figure out the sizes of the fragments. Might be important in identifying plasmids. It might be important in gene therapy, but we're going to talk about the basics, and then we're going to walk through together how to actually figure out the sizes of these DNA fragments. All right, so we're going to start here. So in a restriction digest, the restriction enzyme that you choose is being used to cut DNA into fragments. And those fragments are going to be called restriction fragments. Sometimes they're called DNA restriction fragments. But essentially, we're just cutting up pieces of DNA. You can use many types of enzymes. So for example, we know that there's EcoR1 that was found in E. coli. There's also one called BAMH1 and HIN3. So there are over, I think, 600 commercially produced restriction enzymes that all cut at very specific sections of the DNA, and they're all going to cut at different sections. So you might carry out a restriction digest for multiple reasons. So one of those reasons is that you want to isolate genes. So maybe you're targeting a section of DNA that you then want to insert into a plasmid, put it into bacteria, collect the protein, much like what is happening with insulin. Or you may want to cut the plasmid, right? So we want to make sure that the plasmid we use is also cut by the same restriction enzyme that we use to cut the initial section of DNA. So you can use a restriction digest to isolate genes, number one. You can use the restriction digest to cut the plasmid so that it's ready for recombination. You can use a restriction digest to identify DNA. It's number three. And you can also use this technology to support genome mapping. Because we're using restriction enzymes isolated from bacteria that usually live within people, most of them are designed to work at body temperature, which is 37 degrees Celsius. So that's what that picture is showing, is that you want to, if you're carrying out a lab in which you're using restriction enzymes, the DNA with a restriction enzyme would be incubated at 37 degrees Celsius, which is what we did for our jelly electrophoresis lab. We incubated it during the day one procedure in the incubator for 24 hours at 37 degrees Celsius. Like most restriction sites, the sequence of base pairs is the same whether you read it from left to right or from right to left, and that's because of the way our nucleotides pair up, right? So if you look at this section here, this is where this BAMH1 restriction site will cut. So it's looking for GGATCC on this section of DNA, so it's going to read it this way. And then there's also going to be a restriction enzyme that's going to read it this way. And so it's going to look for GGATCC. But if you look at them, you'll notice that if you read it this way, if you read it this way, they're the same. The letters are in the same order. That's called a palindrome. So when the base pairs are in the order so that if you read them from left to right or right to left, they're the same essentially. And you'll see that again here, GAATTC, and it goes in the same order, just backwards. So like I said, there are more than 600 different restriction enzymes commercially made. They've all been based off of what we figured out bacterial cells were doing to defend themselves from viruses, which is pretty cool. They're all routinely used in laboratories to modify DNA, so for different purposes. Researchers use a particular restriction enzyme depending on the restriction sites that are present in the piece of DNA that they're working with. So there's these 600 restriction enzymes, but how do you choose which one to use? How do I know if I want to use BAM or if I want to use EcoR1 or HIN3 or 597 other restriction enzymes? How do I pick one? So usually you're going to look for a restriction enzyme that cuts at a restriction site that's as close as possible to the end of the gene. So you want to figure out your gene sequence, and then you're going to look outside of that a little bit. So maybe my gene is right here that I want to isolate. I'm going to pick a restriction enzyme that cuts really close to that. I don't want it to cut too close so that it actually damages this gene here, but I want it to cut close enough so that I don't have like a bunch of extraneous DNA that I'll then have to deal with. So the way you choose is by finding a restriction enzyme that has a restriction site, so where it's going to cut, that is really close to your gene of interest. So pretty common sense, I think. If a researcher is trying to isolate a gene after carrying out a restriction digest, only one of the restriction fragments would contain that gene. And so we want to separate them and identify the restriction fragments, and the way we're going to do that is through jelly electrophoresis. And so you should be very familiar with this process. But in jelly electrophoresis, so just in case you need a reminder, also a good review for your quiz coming up, the negative charge that DNA restriction fragments carry and the fragment size are the properties that are used for separation. So we know that the gel box, the chamber, has a positive side and a negative side. We know that we run electricity through it. We're going to push the DNA from one side to the other using that electrical charge. And then we're hanging on to this idea that the smaller molecules are going to move further away from your starting point. So the researcher is going to add the restriction digest mixture to a well in the gel, just like you guys did. The researcher will then turn the electrophoresis box on, and it's going to apply an electric field across the gel. The negatively charged DNA restriction fragments move away from the negative electrode until you stop running the machine. And so essentially they're going to move from the negative side to the positive end, and we're going to separate them out by size. So just like you see here, so the smaller the DNA restriction fragment, the farther it migrates through the gel. And so this blue line would represent our wells. And so we have this really big, huge piece of DNA here that wasn't really cut. Maybe that was our original section. This one's a little bit smaller. You can see the sticky end here. It was cut. Even smaller here, so we're counting the number of bases essentially. And so even these would count, the ones that aren't paired. And smaller and smaller and smaller, and this would be our smallest fragment size, which should be furthest away from the cell. All right, so just to recap, the agarose gel has tiny pores in it that allow smaller DNA fragments to move through it relatively quickly. Larger fragments are going to move much more slowly through the gel. After exposure to electric field, the DNA fragments are separated by size. Fragments that are the same or similar in size will move together through the gel. The speed of DNA movement can be increased by increasing the voltage. However, the higher the voltage, the lower the resolution. So you're not going to get really nice bands in your gel. They're going to be kind of fuzzy. They kind of bleed into each other, and you may not be able to see individual bands when you look at your electrophoresis gel. What you'll get at the end is something that looks like this. Okay, ours were a little bit different, but you can see that these are nice, clear bands that was ran really nice and slow. And so the B, E, and H represent the different restriction enzymes we use to cut the DNA, and so you can see they produce different size fragments. Because the DNA is colorless and can't be seen, stain is added to the gel. So that's what I did with our last gels. The blue color came from the stain. The stain binds to the DNA and does not bind to the gel. So if I were to continue to wash that out, that blue would go away. A group of fragments of the same size will typically appear as a band with trailing edges. So that's kind of what's happening here. You can see they're kind of like fuzzy here. That's because there's multiple fragment sizes that are like really close in size, so they kind of ended up in the same place at the gel, especially this one here. Very large pieces of DNA appear as a smear close to the well. The bands at the bottom of the gel are the smallest fragments because they migrated faster and therefore traveled a larger distance from the well. The cool thing is that if we have a DNA ladder inserted here, so what that means is you have DNA that's been eaten by a restriction enzyme, or cut up, I shouldn't say eaten. It's been cut up by a restriction enzyme, and it's going to produce fragments of a size you already know. So you know the size of the different pieces, so you can go back and use that to create a curve, which you can then use to figure out how large these other pieces are based on how far they traveled. And this is what we're going to work on today. So it's standard practice to fill one of the wells in the gel with a sample of known fragments. So that's usually your DNA ladder. The results from this well are used to plot a standard curve. So that's what you're seeing here. So we're looking at the size of the DNA fragment, which we count in base pairs. So BP means base pairs. And then here at the bottom, you're looking at how far the fragment traveled away from the well. And we're looking at that in centimeters. The graph paper looks a little bit different because this is log paper, so we're going in base tens here. So if you start at 1, you go up to 10. Each line in between represents 1. So this would be 2, 3, 4, 5, 6, and it gets really crunchy as you get up to 10. And then this next group, 10 to 100, each line represents 10. These would each represent hundreds. These would each represent thousands, and you can increase. So it allows us to put all this data on a graph that's nicely, tightly packed, or else our axes, if we had to go from 1 to 100,000, it would be huge. Like I couldn't give you a graph paper big enough for that. Okay, so the results from the well are used, the results from the DNA ladder are used to plot the standard curve, which essentially is a graph of fragment size against distance traveled. The sizes of unknown fragments can then be estimated from the standard curve. So for each unknown band in the gel, you'll carefully measure the distance it traveled from the well, and then you use the standard curve to match the distance to the fragment length in base pairs. So if we go back, let me see if I can. Bad news bears. All right, so if we go back here, like we would know our DNA ladder sizes, but if we wanted to figure out how big a piece of DNA this is, we would take our ruler and we would measure from the end of the well to about the middle of the band in centimeters, and we would say maybe that's like 2 centimeters, and we could look at our standard curve and say, okay, here's 2. I'm going to go all the way up. And where it crosses the line is going to tell you how big, roughly, it's an estimate, how big a piece of DNA you have. So this is, what, 10,000 at this line? So this would be 11,000. So maybe 11,500 is how many base pairs long that piece of DNA is. So the standard curve is really important because it allows you to estimate the sizes of the other pieces of DNA. All right, so the variation in the nature and concentration of the agarose impacts the speeds of the fragment through the gel. So you can get some differences from gel to gel, but if you run your gel nice and steady and nice and slow, you can come up with a really nice standard curve, and you can get really good estimates for the sizes of your DNA fragments, which is what we're going to work on next. I hope this was helpful. I hope you understood what is going on in the graph. If not, we're going to practice together, but this will be on your quiz, so you need to make sure you know what is going on. All right, so we're going to go ahead and stop here. Thanks for listening.